the nucleus and ribosomes. The nucleus is where the DNA is housed. The nucleus also houses subunits of ribosomes. Okay, this particular region of the nucleus that houses the subunits of ribosomes is called the nucleolus. You'll see it if you look at slides of the nucleus as the densely staining body within the nucleus. The nucleus is bounded by a nuclear envelope. The nuclear envelope is not continuous. As you can see over here, there are pores on the nuclear envelope. It's more like a thatched roof instead of a plastered wall. The nuclear envelope is continuous with another membranous structure. This membranous structure is the endoplasmic reticulum. On the endoplasmic reticulum, these dot-like structures are the units called the ribosomes, which I told you earlier are the protein synthesizing units of our cell. In a transmission electron micrograph picture, you can see how the nucleolus is darkly stained and it is housed within the nucleus. Here you see a tangled web of DNA. DNA complexed with protein is called chromatin. The ribosomes are chemically made up of ribosomal nucleic acid or rRNA and protein. So there are two macromolecules that make up the ribosomes. The job of the ribosomes is to synthesize proteins, and they do so according to the instructions they receive from DNA. So if their job is to synthesize proteins, you probably guessed that in a cell that has to make a lot of proteins, there will be a lot of ribosomes. So if you look at a slide under the microscope and you see many densely stained, very tiny bodies all over the cytosol and on the endoplasmic reticulum, you can tell that this cell is, or at the time of making that specimen, was in the process of making lots of proteins. So this is one part of the flow of information. The DNA transmits its mRNA out into the endoplasmic reticulum where the mRNA meets the ribosomes. You see the bound ribosomes are these brown dots on the endoplasmic reticulum, and the ribosome then begins to make protein. Okay. The nucleus, as you saw earlier, is in conjunction with the endoplasmic reticulum, and that endoplasmic reticulum brings us to our organelles, which make up the endomembrane system. The organelles that are part of the endomembrane system all interact to carry out the following functions: synthesis, distribution, storage, and export of molecules. The endoplasmic reticulum, or ER as it is often known for short, is a membranous network of tubes and sacs. There are two kinds of ERs, a smooth ER and a rough ER. And these can be recognized by the fact that smooth ER is smooth and rough ER has ribosomes on its surface. You'll see in this picture. A smooth ER is a smooth membranous system. The rough ER, as you see here, has those brown dots that I said were the ribosomes. Again, you can see the smooth ER out here too. It has no dots on its surface, no ribosomes on its surface. Why do these two endomembranous systems exist? Well, they exist because they carry out two different functions. The smooth ER is responsible for synthesizing lipids and for processing toxins. The rough ER is what produces membranes. It has ribosomes on its surface, so it makes all kinds of proteins, membrane and secretory proteins. This is a diagrammatic view of the smooth and the rough ER. Okay. Here you see synthesis and packaging of a secretory protein by the rough ER. The rough ER is rough because it has a ribosome in it, and this is a ribosome. The ribosome has received a message from the DNA in the form of mRNA. It's decoding that message as the mRNA passes through the ribosome, and while it's decoding, it is carrying out the the job it's supposed to. It's making protein, and here's a long growing polypeptide that's going to make that protein. That polypeptide is housed within those membranous structures that we saw that make up the rough ER. In this particular diagram, you see the protein is getting modified by adding sugar residues. Sugar chains are being added to the protein, and therefore this protein gets called a glycoprotein. Glyco for sugar. The glycoprotein, when it's completed, its manufacture is completed, gets ready to be transported to the rest of the cell. And that happens by this glycoprotein moving to the end of a membrane system where it can get pitched off and form its own little secretory vesicle. And this vesicle then is then responsible for taking the glycoprotein to its ultimate destination. The other organelle that's part of the endomembrane system is the Golgi apparatus. The Golgi apparatus consists of stacks and sacs, again, like the ER. And in the Golgi apparatus, the products that come out of the ER, that's the proteins, are further processed and then sent to other organelles or to the cell surface. Okay, so here this diagram shows you the connection between the ER and the Golgi. The brown-colored, brown painted organelle here is the Golgi. It has just received that transport vesicle that we saw in the earlier slide that is housing within it the glycoprotein. The glycoprotein then fuses with the Golgi apparatus on one end, so there is a direction to functioning of the Golgi apparatus. The end of the Golgi apparatus that receives that transport vesicle is called the receiving side of the Golgi apparatus, and the opposite side is called the shipping side of the Golgi apparatus. So the vesicle containing the glycoprotein fuses on one end. It then gets modified in the Golgi apparatus the way it needs to in order to become a functional protein, and then it comes out from the other side in the form of a vesicle again. And onto the shipping side of the Golgi. The other organelle within this function is called the lysosome. The lysosome houses enzymes that break down ingested substances and damaged organelles. So here you see, for example, food trying to enter into the cell. It can only do so by an invagination of the plasma membrane. The invagination of the plasma membrane results in this organelle, results in this food particle being enclosed in a sac-like environment made up of plasma membrane. This thing is called a food vacuole. The food vacuole is ready to fuse with the lysozymes, because remember the lysozymes have enzymes in them. When the two fuse, this becomes a phagolysosome. And this new structure now is what allows the newly ingested food particle to be completely broken down because of those enzymes that came from the lysosome. Okay. Here is another example of the function of a lysosome. Here is a vesicle that contains damaged mitochondria, a mitochondria that has lived its life. Remember, everything has a half-life. If you recall, I did talk about autophagy in an earlier lecture. This is the process by which the cell eats up some of its own components when those components have lived their life, or the cell needs energy in some form that it is not able to get from the outside. So this damaged vesicle also can fuse with the lysosome. As you see here, the lysosomes then functions in digesting all the contents of that new vesicle that it has fused with, and the components of that digested vesicle are released into the cell for the cell to use or reuse more appropriately. Alright, another organelle that you will see in some cells is called the vacuole. The vacuole is a large vesicle and it can have various functions. In some protists, remember protists are the unicellular but eukaryotic cell type. Some protists have contractile vacuoles. As the name suggests, a contractile vacuole can contract or expels its components after the cell contracts. Plant cells have something that we call a central vacuole. It stores molecules and waste and facilitates growth. You won't see a central vacuole in an animal cell. This is a unique characteristic of plant cells. Okay, here's a light microscopic image of a paramecium, which is a unicellular eukaryotic, a unicellular eukaryote, and you'll see contractile vacuoles within this organism. An example of a central vacuole that you'll see in a plant cell. We'll see all these when we look at slides this afternoon. Okay. What about the organelles that are, so the next slide is a review of all the structures that are involved in manufacturing and breakdown. Okay, so there are endoplasmic reticulums that form transport vesicles. They fuse with Golgi apparatus. The Golgi apparatus then is responsible for ultimately releasing the proteins that have been synthesized out into the cell. One more structure that is involved in the breakdown or manufacturing of molecules, one more organelle, is the peroxisome. Okay. The peroxisome is listed separately here because it did not originate from the endomembrane system. That's